part of it, so maybe give your brains a little chance to rest. So the good part is, again, not a lot of detailed, scientific concept to get at, but there's a lot of vocabulary. So I'd say that the challenge of this lecture and the next lecture will be the vocabulary that you have to kind of get straight and learn from. So some of you guys might already be well versed in this vocabulary. I know I still get confused about some of the words that are very similar to each other, but I'll point that out. So again, some of you guys might like this a little bit more, some of you guys might find this boring. Either way, it's a little bit of a change in direction from where we've been the last couple lectures. Okay, so epidemiology is really the study of disease, not from a scientific aspect as much, but where the disease comes from, the distribution of the disease, the frequency of a disease, what causes the disease, not just what, this strain of bacteria causes this disease, but are there certain types of people that are more likely to get that disease? Is there a region, a geographical region where more people are likely to get this disease? So it's, again, more of the social aspects of the disease than as opposed to just the scientific part. Okay, so here's some kind of famous epidemiology studies that have been in the past. So in the mid-1800s, John Snow was the first one to notice that the occurrence of cholera in London was following some kind of pattern that he thought he could decipher. So he actually mapped the occurrence physically on a map, drew out where there were pockets of people that were getting it versus pockets of people that were not getting it. Okay. Around the same time, Semmelweis showed that hand washing decreased the incidence of fever. So this is, again, something we take for granted now, but back then people didn't really know the concept of germs. They didn't think hand washing was going to do anything except get dirt off your hands. They didn't realize there was anything to infection about that. But he actually showed that hand washing decreased the incidence of puerperal fever. And then Florence Nightingale, a lot of you guys probably heard the name before. A lot of you guys probably don't know exactly what she did. But one of the major studies that she did was that she showed that improved sanitation, so including hand washing, but a little bit more than that, decreased the incidence of epidemic typhus. Okay? So there's really three different types of epidemiology studies like these people did. So there's descriptive, which is basically looking backwards in the past, looking at the pattern of an infection and trying just to collect, analyze data to determine what might have caused that infection. So it's kind of looking in the past. It's not actively doing an experiment, it's just collecting data and trying to make a pattern out of it. So again, Snow, who just mapped the occurrence, again, he was just mapping it. He wasn't setting up an experiment. He was just kind of looking at the natural progression of the disease and just collecting data on it. There's analytical, so that would be taking a disease group and a healthy group and comparing the two. So Florence Nightingale, again, she took a group of people that had good sanitary habits and people that had poor sanitary habits. She didn't set up the experiment. She didn't say, hey, you guys shower and you guys don't, and I'm going to see who gets epidemic typhus. But she just looked at people that got typhus and then observed them to see if they had good sanitation, looked at people that didn't get typhus and observed them to see if they had good sanitation, and then made the conclusion that the people that were cleaner were not getting typhus as often. Okay? So it wasn't something she set up again, it was something she just observed, but she looked at a control group versus an experimental group. Okay? Semmelweis was the final kind, which is an experimental type of epidemiology, which is actually putting yourself in the experiment and setting up a control and experimental group and seeing what happens. So Semmelweis, he actually took people and said, I want you guys to wash your hands every day. I want you guys to never wash your hands. And I'm going to come back in 3 months and see how many of you guys got this puerperal fever. And what he found out were the people that he told to wash their hands were not getting the fever as much. So he actually set up the experiment. It was a little bit more than just observing. It was actually setting up a control group, an experimental group, and then collecting the data and making a conclusion. Okay, so epidemiology. A big part of epidemiology is case reporting. So healthcare workers, if there's an outbreak of a disease or maybe a food poisoning, they report these cases either to the state level or if it's big enough to the national level. Physicians are part of this, so if they see, again, a spike in a certain type of, patients with a certain type of disease are coming in more frequently than they normally do, they are required to report this to, again, either state authorities or national authorities. So again, if it were something like, What's a good example? What's the one with the spores? I can't think of it right now.
Anthrax?
What's that?
Anthrax?
Anthrax, yeah. So if there's just a general physician who never saw anybody with anthrax, and then in the week 10 people came in with anthrax, he'd report that straight to the national level. Okay, that's a huge deal. Okay, so the governmental agency that's kind of collects this data and does research around this data and gives advice to people is the Center for Disease Control and Prevention, the CDC. So they collect and analyze this epidemiological information in the U.S. They publish a pamphlet, let's call it, called Morbidity and Mortality Weekly. Some light reading for you guys. Basically, it gives you data for that week about a variety of different diseases and if there's any outbreaks going on anywhere, and then suggestions around that. If anybody's interested, here's the website. Okay, so here's kind of the first set of vocabulary that we'll get into. So morbidity versus mortality. Okay, morbidity is basically just the incidence of a disease. Okay, so morbidity would be the morbidity rate of the flu would be everybody that has the flu. Okay, mortality is specifically deaths. So the mortality rate of the flu would be anybody that died from the flu. Okay, so morbidity rate is always higher than mortality rate, unless the disease kills everybody that gets it, in which case they would be equal to each other. Okay, pathology, and we'll be talking more about pathology on Wednesday, but it's the study of disease, just very generic. Etiology is the study of the cause of a disease. Infection is the colonization of the body by pathogens. And the actual definition of disease is an abnormal state in which the body is not functioning normally. Okay, it's a very generic term. Okay, so we'll talk about normal microbiota. We've talked about it before. So there's a couple different types of normal microbiota. There can be transient microbiota, which may be present in the body for just days, weeks, or months, but normally isn't present in the body, but it's not harmful. So it's not causing a disease, but it's not normally found in the body. Normal microbiota is it permanently colonizes the host, so it's constantly found. So E. coli in the intestine, everybody has it there. That's normal microbiota. Symbiosis, and we'll talk about that, is the relationship between normal microbiota and the host. So here's some examples: bacteria in the large intestine, nasal epithelium, and in the lining of the stomach. So here's different types of symbiosis. So symbiosis is just the relationship. Now that relationship can take many forms. Commensalism is when one organism benefits and the other is unaffected. Okay. Mutualism, which most normal microbiota relationships are mutualism, where both organisms benefit. And there's parasitism, whereas one organism benefits at the expense of the other. Okay. So most of them again are mutualism because, for instance, E. coli in the gut has some positive effects that we'll talk about, but then it's getting nutrients from the food we intake. So they get some benefit from us, but we get benefit from them also. So here are the more common places where you find normal microbiota in the human body. In the mucous membranes, throat, eyes, nose, mouth. On the skin, we all have different strains of staph-type bacteria on the skin. In the intestine is where you have most of your normal microbiota, and then in the urinary and reproductive systems. In specific places, there are places in the reproductive systems that are completely sterile, and if you do get an infection in those areas, it's a big deal. So here's, again, a little bit more about microbiota. So microbial antagonism is the competition between microbes. So that's kind of how typically the normal microbiota we have helps us. It basically antagonizes pathogenic bacteria, so bacteria that causes disease. It basically prevents bacteria that causes disease from being able to colonize our body because the normal microbiota has already colonized it. So it just physically takes up space to prevent colonization by bacteria that can harm us. So it occupies niches that other pathogens might occupy. They also can actively produce acids or other chemicals that kill off other bacteria. And so some of you guys, have you guys heard of probiotics? Like in yogurt, they talk about it. Obviously, it's the opposite of antibiotics, so it's something that promotes bacterial growth rather than inhibits bacterial growth. So again, it's, if someone has lacking normal microbiota, they give probiotics to help that bacteria grow, because again, it's essential to have this normal microbiota in your system to prevent pathogens from colonizing your body. So there are certain cases where your normal microbiota, where a normal situation is completely not harmful, it's actually beneficial to you. There are situations where they can become opportunistic pathogens. So basically these bacteria which would not cause harm in a normal situation can start to do so. These are the main ones. So immune suppression. So our immune system is keeping this normal microbiota at basal levels, basically preventing it from overgrowing. If for some reason your immune system is not functioning properly, the normal microbiota can start to kind of take over a little bit and can become a pathogen, can cause disease. If there's a significant change in the normal microbiota, that can sometimes lead to disease. And sometimes normal microbiota that is completely innocuous in one site, if it accidentally gets into another part of your body, can become a very big deal. So E. coli that's in the large intestine is not a big deal at all. If it got into your bloodstream, that could be a pretty big deal. So again, normal microbiota has to be in its specific niche in your body in order to be not harmful. And if it somehow gets out of whack, it can become harmful. Okay, so this is probably one of the more significant portions of this lecture, Koch's postulates. Basically, it's a way to figure out what organism causes a specific disease. Okay, so there's a new disease, nobody's seen it before. They want to figure out what organism is causing this disease. These are the type of questions that scientists ask themselves when they're trying to figure that out. So basically, if all four of these are true, you can be fairly sure that you've figured out what pathogen causes a specific disease. Okay. So number one, the same pathogen must be present in every case of the disease. Okay, so if you have 20 people have this disease that you've never seen this disease before, and you take some blood from them and you isolate the bacteria from them, all 20 of those patients have to have the same bacteria. If only 19 of the 20 do, then you have to keep digging. Okay, so every single patient, every single case of the disease has to have that pathogen. Okay, so that has to be true, number one. Number two, the pathogen must be isolated from the host and then grown in pure culture. So you've isolated the bacteria from all 20 of these people. You've done some tests to figure out what it is. Now you have to grow them in culture. And when you do grow them in culture, you have to analyze them after they've been grown in culture and confirm that they are all the same. You take the pathogen from this pure culture, and then you inoculate a healthy, susceptible lab animal with this bacteria, and every lab animal has to get the same disease. Okay? And then you basically do that again. After those animals get that disease, you take some of their blood, you isolate the bacteria from them, and it has to show that the bacteria that actually caused the infection in those animals is the same exact bacteria that caused the infection in the original humans. Okay? Now, if all four of these things are true, in most cases, you can say with 100% confidence you figured out what bacteria or virus has caused this specific disease. If any of these are false, you're maybe close, but you're not completely right. Okay? So here's just a graphical definition. So in this case, they're finding a disease in mouse. They take microorganisms from the mouse, they identify them, and they grow them in pure culture. They take the bacteria from the pure culture, they inoculate a healthy mouse. Okay, that mouse now basically has to die in order to ensure that they got the right bacteria. If it does, they isolate the bacteria from the second dead mouse, and it has to be the same as the bacteria from the first mouse. And if all these things are true, then you can be pretty sure you figured out what caused that disease. So there's a couple of, what do you want to call them? Problems, issues with doing these tests. So there are two pretty big ones that can cause this to be confusing. So one is some pathogens can cause several disease conditions. So a single bacteria strain can cause a different form of the disease in various people depending on their immune system and their genetic makeup. So you could have 10 people that all have the same disease. You take the bacteria from them and you inject, let's say there was nothing ethically wrong with this. You inject a healthy human and they get a slightly different disease. Okay, so you would say by Cox postulates, you would say, oh, I didn't identify the right bacteria. However, you might have actually identified the right bacteria. It's just this person has a slightly different immune system that caused the disease to look a little different to them. Okay, so that's one issue with these tests that you have to be wary of. The other problem that I kind of just referred to is that some pathogens only cause disease in humans. So again, based on Cox postulates, you take the bacteria from the infected human, you isolate it, and then you infect a lab animal to see if the lab animal gets the same disease. Well, if your disease is only specific for humans, your mouse might not get the disease, and you might think, oh, I didn't isolate the right bacteria, but it might really be only because that bacteria doesn't cause disease in mice, it only causes disease in humans. So something like HIV, when they first discovered HIV and AIDS, they isolate this virus, all these people have this virus, we think this is causing AIDS, and then they inject it in a mouse, and the mouse is fine. And they thought, oh, well, we didn't find the right virus because the mouse is fine. In reality, it's just HIV doesn't cause AIDS, doesn't cause immune failure in mice, it only causes it in humans. Okay? So that's a problem with this because you can't, again, like I said before, ethically you can't just go infect healthy humans and see if they get the disease. You have to use lab animals, and sometimes a certain disease doesn't carry over to lab animals. Okay? So these are two things you have to be wary of when you're using Cox postulates. Okay, so here's a bunch of different vocab type terms that we'll go over. So when you classify infectious diseases, so a lot of you guys probably already know this, or you might have some idea about it, and this will just kind of help enforce it. So a symptom is a change in the body function that is felt by a patient as a result of a disease. So a symptom would be fever, chills, that's a symptom. So that's something the patient actually feels. Okay? A sign is a change in the body that can be measured or observed as a result of the disease. So the difference is a symptom is a sign, but a sign could also be something the patient can't feel. Okay? So if someone has, again, just because I'm most familiar with it, if someone was infected with HIV, they might feel okay for a while, but if a doctor did a blood test and saw that their white blood cells were very low in numbers, that would be a sign that maybe they have HIV. It's not a symptom because it's not felt by the patient, but it is a sign that the doctor would be able to tell. A syndrome is a specific group of signs and symptoms that accompany the disease. So again, a syndrome for the flu would be like chills, sweating, fever. They're all the symptoms that most people have. If you put them all together, that would be the syndrome. Okay, here's another one that I didn't know until I started teaching this course. I'd heard them, but I didn't know the difference. So there's a communicable disease versus a contagious disease. Okay, communicable disease is a disease that is spread from one host to another. A contagious disease is a disease that is easily spread from one host to another. Does anybody can anybody give an example of each one of those, or think that they might know what the difference is by giving an example? I know it's not described very well here, but think about it for a second and see if you can come up with an example.
AIDS.
A communicable disease being mono HIV.
What was the first one?
Mono?
So I think mono would fall closer into contagious because it's fairly easily spread. HIV is a good one for communicable because it can be spread from one person to another, but not very easily. So usually, it's kind of a fine line of what you consider easily. Mono you can get by fairly simple touching, so that's closer to the contagious disease, whereas HIV takes a little bit more significant contact in order to spread from one person to another. So the ones I usually use are HIV, and then the flu is contagious. You can get it just from someone sneezing in the same room as you can get the flu. Okay, non-communicable disease is a disease that is not transmitted from one host to another. Anybody come up with an example of that?
would be from, let's say, a human host to an animal host that it would harbor the disease?
Yes, I mean, that's true. That's true, but even within one species, there are non-communicable diseases. So, I mean, the one that is, comes to mind first is something like cancer, right? Completely non-contagious and non-communicable. It doesn't go from one host to another. All right, so a couple more. I know there's a lot of these definitions, but they're important in order to set up the rest of the course here. So incidence versus prevalence. Okay, so the incidence of a disease is the fraction of a population that contracts a disease during a specific time. So if I said, what's the incidence of, or how many people got the flu in the last 3 months, that would be the incidence. So I've given you a specific time frame, how many people got the disease in that time frame. The prevalence is how many people have that disease right now, or how many people had that disease on February 27th. Okay, so at a specific point in time, how many people in the country or how many people in the world have that disease? That's prevalence. Whereas again, incidence is over the last year, how many people contracted HIV. That would be incidence. Okay, so again, incidence, how many people got the disease in a certain amount of time. Prevalence, how many people have that disease at a given time. So 1 million people contracted HIV in the last year is incidence. Right now in the world, 10 million people are HIV positive. That's prevalence. Okay. Sporadic disease is disease that occurs occasionally in the population, so it's not seen year-round. So again, the flu is one that falls into sporadic. It's not around typically for about half the year. Endemic disease, on the other end of the spectrum, is something that's constantly present in the population. At any given time, there's someone somewhere that has this disease. Typically not just one person, but many people. So chickenpox. At any given time, if you talk about how many people in the country have chickenpox, there are always a significant amount of people that have chickenpox at any given time. It's never gone. Okay. An epidemic disease is a disease that's acquired by many hosts in a given area in a short time. So again, that'd be something that typically is a little bit out of character. So if we had a flu, I don't know the right number, let's say typically 10 million people get the flu every winter, and for whatever year, this year 100 million people got it, that would be an epidemic because it's acquired by many more people than you would usually see in a given time. Pandemic is an epidemic, but it's worldwide rather than in a specific region. And then herd immunity is immunity in most of a population. So there are small clusters of people in various places throughout the world that kind of breed within their own groups, and they have very similar genetic information, and so they have this herd immunity where the entire colony of people is basically immune to a certain disease, where the majority of the people in the world are not immune to it. Okay, so switching gears a little bit to the severity or duration of disease and words you would use to describe that. So an acute disease are when the symptoms develop rapidly. So again, something like the flu, you get infected within 48 hours, you usually have pretty severe symptoms, comes on quick, typically goes away quick. I know it doesn't feel quick when you have it, but in the scheme of diseases, it's fairly quick, usually within 7 or 10 days it's been removed from the body. Chronic disease is different from that. It's a disease developed slowly. Typically, it can't be felt very significantly in the beginning, and as things slowly go along, and time goes on, it gets worse and worse and worse and worse. These tend to be more severe, the chronic diseases. Subacute disease is basically a combination of the two. An acute disease can become chronic if it's not treated. And so in that period of time when it's transitioning from acute to chronic, you call it subacute. And then a latent disease is something we've talked about. Again, something like the herpes, where you have an acute disease, it goes away for some amount of time, and then something triggers it, comes back sometimes years later. That would be a latent disease. So there's no symptoms while the disease is gone, and then it comes back and you get the symptoms again. So again, there's different types of infections. Local infection, where the pathogens are limited to a small area of the body. That would be like a rash on your arm, let's say. That would be a local infection. Systemic infection is typically more severe. It's when you have an infection throughout the whole body. Focal infection is when you have a local infection, again, that's not treated correctly, and it becomes a systemic infection. Sepsis is a term a lot of you guys have probably heard. It's an inflammatory condition arising from the spread of microbes from a focus of infection. So that's in the transition of focus to systemic. You suffer from sepsis. Basically, your whole body is fostering an immune response to something that started as a local infection. It's now starting to spread to the rest of your body, and you get this overwhelming inflammatory response across your whole body, and that causes people to be very ill. Bacteremia is bacteria in the blood. Septicemia is the growth of bacteria in the blood. Toxemia, toxins in the blood. Viremia, virus in the blood. Those are pretty self-explanatory. Again, primary infection is that first acute infection that causes the initial illness. A lot of times you have a primary infection and that's it. Sometimes you have a primary infection, it starts to go away and something happens and makes it come back. Has anybody ever had that? Where they're sick, they start to feel better, they think they're all better, and then a few days later they start to feel sick again, it comes back. A lot of times it's worse than it did the first time. It's a secondary infection. Sometimes it's the same disease coming back. Sometimes it's you get the primary infection, your immune system has a hard time getting rid of it. It does eventually, but it basically takes up so much of your immune system that you get a secondary infection from another microbe because your immune system is spent so much time trying to fight off the first infection. Subclinical disease is no noticeable signs or symptoms. So inapparent infection, so someone that has subclinical disease would not necessarily know they have the disease because they don't feel any symptoms or signs. All right, we will stop there. Finish up this on Wednesday, and we'll go into the pathogenicity lecture also on Wednesday and finish both of those up. Okay? Any questions about this or anything else?